Bismillahirrahmanirrahim. We start with the paper 2, which is this is a March 2021 paper 2, which has just the results have just been declared last month, and this is a very latest paper, and we are going to discuss this in maybe two videos because there are about 6 questions, and maybe we can only do 3 questions in one video. So let's start. Let's read through question number 1, which is figure 1.1 is an image of a transverse section through the trachea of the gas exchange system. This is the lumen of the trachea. This is the ciliated epithelium which lines the trachea, and then we have the perichondrium, and then we have to label X and Y. Now, X was the mucus gland, and Y was the cartilage. You see, the cartilage has these typical structure which you can see here, and they have these cells with these sort of nucleuses, and you need to be really clear when you have to pick up any of these photomicrographs. So, cartilage. Then it says write the names of the structure X and Y in the boxes that provided. So that was for 2 marks. Please always look at the marks. Then the perichondrium contains collagen fibers composed of collagen molecules. Collagen is an example of one type of biological molecule. State the name of the type of biological molecule of which collagen is an example is fibrous protein. You studied this in chapter 2, fibrous protein and globular proteins. If they had asked you maybe hemoglobin, then it would have been a globular protein. So the two different types of protein you must know, fibrous proteins and globular proteins. Then it says some structures in the gas exchange system are listed in alphabetical order in table 1.1. Write yes in the box provided of the structure which contains smooth muscles, and no in the box provided the structure does not contain smooth muscle. So alveolus does not contain any smooth muscle, but the trachea, bronchus, bronchiole all contain the smooth muscle. So that was 1 mark, but you had to get all this correct. This whole thing had to be correct to get 1 mark. Even if one was wrong, well, you'd get a 0 because there's no 0.5 in biology. Then let's start with question 2. Starch molecules are the main storage molecules in many types of cereal grain, such as the grain of the barley plant. When the seed inside a barley grain germinates, genes coding for digestive enzymes are switched on. The enzymes that are synthesized catalyze the hydrolysis of storage molecules such as proteins and starch. Explain what is meant by a gene. Gene is a length of DNA, or you could say the sequence of bases, sequence of nucleotides, and which codes for a polypeptide, or you could have said a protein. Now this is 2 marks, and you have to give me 2 points. So always think when you look at the marks, always think of the 2 points. So length of DNA or the length of a sequence of nucleotides or sequence of bases, nucleotides better, which code for a polypeptide or a protein. You see, it's like AT, AT or GC or CG. So these bases code for a polypeptide or a protein molecule because they code for the sequence of amino acids. So they code for the primary structure of the amino acid. And then everything is automatic. The folding is automatic because the R groups then bond and form the hydrogen bonds or the ionic bonds. Then coming to part 2. Now when you look at part 2, it says the hydrolysis of proteins in the barley seed produces amino acids that can be used in the synthesis of the proteins required for the formation of the seedling or the young plant. Seedling, duck, baby, duckling, seedling. We must know. Some of you are a little weak in the English part of this, so please improve your English. That is one of the biggest problems that we face whenever we have students which struggle with the English part. Figure 2.1. Figure 2.1 is an incomplete diagram of the molecular structure of the smallest amino acid, glycine. Each molecule of glycine has 2 carbon atoms. Complete figure 2.1 to show the molecular structure of glycine. I've just done that for you. N and H, there was a minor part had to be added, and then the carboxylic acid part, C double bond O and then COH. Starch is a mixture of 2 different molecules. Name these 2 molecules. Now we all know this. That's basic recall. Amylose and amylopectin. If you wrote both of them, you got this 1 mark. Otherwise, as I said before, there is no 0.5 in biology. Then we read on the second part of the question. 2 of the enzymes synthesized by the barley seed are alpha amylase and maltase. So they've given you two names of enzymes, and these are involved in the hydrolysis of the stored starch during seedling formation. So the seed contains these two enzymes would be required for the as the seed germinates and these seedling starts to form a little baby plant. In the food industry, the starch extracted from barley seeds, barley starch, is used in the production of sugar syrups. Figure 2.2 summarizes the reaction catalyzed by alpha amylase in the production of maltose syrup and by maltase in the production of the glucose syrup. Now you can see barley starch and it is being acted upon by alpha amylase and results in the formation of maltose. Maltose, you know, is two glucose units joined together and that is called maltose. It's a disaccharide. Both are glucose and this is joined together. It's called a disaccharide. So two glucose units joined together, one glucose here, this would form a glycosidic bond between them, and this would be a maltose. And then the maltose is broken up, so then this bond breaks up, and we have two glucose units separated. So here this glucose is separate and this glucose is separate. So the enzyme maltase breaks down this disaccharide maltose into glucose. Then as we read along the question, some of the substances shown in figure 2.2 are listed in table 2.1. Complete table 2.1 to identify which of the term polysaccharide, monosaccharide, and macromolecule apply to each of the substance listed. Use a tick if the term applies and a cross if the term does not imply. Put a tick or a cross in every box. So you couldn't have left it. Now, as we say, glucose is a polysaccharide. No, it's not. It's a monosaccharide. It's not a macromolecule. Then maltase is an enzyme, so it's not a polysaccharide. It's not a monosaccharide. It's a macromolecule. Then maltose is a disaccharide, so it's not a polysaccharide, it's not a monosaccharide, and it's not a macromolecule. Starch is a polysaccharide, yes, it's not a monosaccharide, and it is a macromolecule. So of course there had to be four ticks and the rest of them were all crosses, and you had to get these lines correct to actually get your 3 marks. So these 3 marks were difficult to get, and we have in the mark scheme how we were going to grade it, if how many correct, how many marks we would give you. When producing sugar syrups, there are advantages in using enzyme extracted from microorganisms. Microorganisms means bacteria, fungi, yeast. For example, some enzymes extracted from microorganisms are heat stable. Heat stable enzymes are used to increase productivity because the reactions can be carried out at higher temperatures. Heat stable means that they are not denatured, they're denatured at very, very high temperatures. So that means they're heat stable. Suggest one other advantage of using enzymes obtained from microorganisms rather than enzyme extracted from barley seeds in the production of sugar syrup. Well, of course, it's easier to extract from microorganisms, and then it's easier to grow microorganisms, to culture them, just culture them on a petri dish, and they can produce large amounts of enzymes. Figure 2.3 is a graph showing how the activity of alpha amylase extracted from barley seeds changes as the temperature increases from 10 to 66. 10 degrees to 66 degrees Celsius. So you have the graph there and explain the effect of temperature. Now that was very basic. What you had to understand was that explain the effect of temperature on the activity of alpha amylase extracted from barley seeds as shown in figure 2.3. Increase in temperature increases kinetic energy of enzymes, so increased rate of collisions, collision between enzyme and substrate, and denatures at temperatures above their optimum, and which of course results in the breakage of the hydrogen bonds. So please remember, whenever it's an explain, you have to give me the biology part of it. But if it's a describe, then there is no biology, and you just have to give me the description of the graph. So that is all what you have to do. Then it says sketch on figure 2.3 the curve that will be obtained using alpha amylase that is heat stable. So naturally the optimum of it would be higher and then the activity is higher at 100% temperature. This was high at this temperature. This was high at this temperature. So you have to understand is how this was graph you had to draw was you'd have to, and there are how many marks for this graph? There were 2 marks for this graph. So one is how would you show the first part of the graph and the second part of the graph. And this is what you had to really show it. You had to show the optimum and then you had to show it decreasing. So this was the graph that you had to draw on this figure 2.3. Then let's look at question number 3. Now question number 3 says T helper lymphocytes and Leydig cells are two types of mammalian cells. So first read the question carefully and understand we're talking of T helper lymphocytes and Leydig cells. The main role of the T helper cell lymphocytes and Leydig cells is to synthesize and secrete cell signaling molecules. Both lymphocytes and Leydig cells synthesize cell signaling molecules. T helper lymphocytes synthesize protein known as cytokines, a chemical known as cytokines, which are protein in nature. Then Leydig cells synthesize the steroid lipid hormone testosterone. So steroid lipid hormone testosterone from cholesterol, and Leydig cells also synthesize cholesterol. So actually Leydig cells are in the testes, and they synthesize steroidal hormones, testosterone. So this information is given to you in the question. Then it says state one way in which cytokines are involved in an immune response. Naturally, cytokines stimulate the macrophage response, they stimulate the B lymphocytes, they stimulate the killer T cells. So it was only 1 mark, and you had to give me all this information. Any one of these would be fine. Figure 3.1 shows part of a mammalian cell. Now you can see this is the nuclear membrane which they're showing here. So you can see a lot of detail of the internal thing of the cell. It's not a sort of a surface view like a scanning electron micrograph. It's not a surface view. You can see the details of the cell, like you can see inside the cell. And inside the cell, you can see there's a lot of this SCR, all these little circles here. This is all the SCR. So smooth endoplasmic reticulum, and of course we can see some of the mitochondria. Then it says state with reasons whether 3.1 shows part of a Leydig cell or part of a T helper lymphocytes. You see, a helper lymphocytes produces cytokines, which was a protein. So any cell that produces a protein should have more RER. But this we didn't see any RER. We're saying Leydig cells a high proportion of SCR. And of course, SER synthesizes the steroidal hormone. So no RER, because RER is needed for protein synthesis. So you could have said these 2 points were also acceptable, and these 2 points were also acceptable for you to get your 2 marks. And this was 2 out of 2, and you would be definitely to get your 2 out of 2, which you should always think when you're thinking of the answer, how would you get the full marks of that part of the question? Then the part 2 of the question was underline the correct name for the type of image shown in figure 3.1 and explain your choice. So was it a photomicrograph? Was it a scanning electron micrograph? Was it a transmission electron micrograph? And as I said, the transmission electron micrograph shows us the details because it's sort of transmission means through and through. So explanation was, this was of course the 1 mark which you had to get to in order to be able to underline this part, transmission electron micrograph. And explanation was cell ultrastructure can be seen at high resolution. It's not a thin section, doesn't show you the surface contours. So there were 2 marks. One was for this. This was necessary. So 1 mark was for underlining this because it says underline the correct name. So there was 1 mark for that, and then for any one of this explanation, there was 1 mark. So that is how this question would have been marked by the examiners who are going to check this paper. Then we come on to the part C of the question. Testosterone molecules and cytokine molecules are transported in the circulatory system to reach their target cells. Now this is a cell, and you know the outer side is a phospholipid bilayer. And you know what cannot cross a phospholipid bilayer and what can cross. So it's fat soluble molecules can pass through it. Alcohol can pass through it. But what cannot pass is small uncharged molecules. Small uncharged molecules can pass, but except the polar molecules cannot pass. So one way in which testosterone molecules could enter their target cells, it says testosterone molecules are able to enter their target and bind to receptors within the cytoplasm. So the testosterone molecule, say it is this one, is going to enter through and through and maybe bind to something here. Maybe something here is going to bind to a receptor inside. It said bind to receptors within the cytoplasm. So simple diffusion down a concentration gradient and can pass through the hydrophobic core, which is the fatty acid tails, because it says is able to enter their target cells. So it is because testosterone would be allowing it would pass through the phospholipid bilayer, would pass through the hydrophobic part because it was a small uncharged molecule or was fat soluble. Number two, cytokine molecules are not able to enter the target cells. Now, that's the reason why they can't enter the target cells. Suggest and explain why cytokines are not able to enter. Yeah, because they're water soluble, they're polar, so they cannot cross the phospholipid bilayer. The phospholipid bilayer is made of phosphate head and fatty acid chains, and this is the area which is hydrophobic. So something which is water soluble and polar will not pass through it. So this is what you had to give me in the question, is that these will be water soluble and polar, so they cannot cross the hydrophobic part, and they will, this is the ones which cannot cross it. So cannot cross the hydrophobic core, too large, and there is no transport protein for them. If there was a transport protein for them, well, then they would pass through that transport protein, but there is no transport protein for them, so they cannot pass. Coming to question 4, using a light microscope and at a magnification of x400, it is possible to identify different types of blood cells and prepared slides of mammalian blood. Figure 4.1 is a key to identify different types of blood in prepared slides. In figure 4.1, letters C, D, E, and F represent 4 different types of blood cells. So now 1A nucleus present, go to 2. 2A large, 1B nucleus absent, that is C. So nucleus absent can only be a red blood cell. So this is what I gave you, red blood cell. Then it says nucleus present, go to 2, large round spherical nucleus. Now that is D, and D was a lymphocyte, large round spherical nucleus. And if present the nucleus not rounded, go to 3, nucleus is kidney shaped, monocyte, nucleus is lobed, neutrophil. So you had to figure this D, E, and F and C, right? Now it says explain why blood taken from a person with an infectious disease may have a different number of white blood cells as compared with blood taken from a healthy person. Naturally, if he's had an infectious disease like he's had tuberculosis or he's had cholera, now when the bacteria enter, then they cause a response, an immune response, and this results to increase in B cells and T cells, B lymphocytes and T lymphocytes, and B lymphocytes produce antibodies. But if you were going to talk about AIDS, then of course the white blood cells would decrease. So if there was another possible answer was also that if you wanted to talk about AIDS, then of course you would say because it destroys the helper T cells, and of course the number of white blood cells decrease. So that was also a correct answer, but I'm mentioning it, but I haven't written it here. As part of a research project, a team of doctors and scientists carried out tests to investigate the effect of altitude, height, on the ability of blood to carry oxygen. They assessed the changes in the oxygen carrying ability of their own blood as they walked from 1530 meters above sea level to 5,700 meters above sea level over a period of 14 days. And 5,700 meters above sea level is an extremely high altitude. Like, for instance, in Pakistan, when we go to Murray, it's at a higher altitude. So, table 4.1 is a summary of some of the results. Table 4.1 shows you that percentage saturation of hemoglobin with oxygen in blood leaving the lungs was 95% at day 1. But when we on day 15 moved to 5,700 meters, it became 82% saturated. The hemoglobin was only 82% saturated, while on day 1 it was 95% saturated. So percentage saturation of hemoglobin with oxygen in blood leaving the lungs. Now, it should actually be 100%, but it was 95%. That's fine. Some of us have it just maybe between 95 and 99. So 95%, but on day 15, it had decreased by 13% to 82%. So you always minus this and you see this is a 13% decrease. Then volume of oxygen delivered to tissues by 100 cm cube of blood. So the volume of blood was the same, 100 cm cube, but it delivered 19.3 volume of oxygen, 19.3, and then it delivered 19.2. So the volume of oxygen also decreased by day 14. So compare the results for day 1 with the results for day 14 in table 4.1. Compare and explain the results. So first of all, there's 1 mark for the comparison, and then the rest of the marks are for the. So if you look at the question, there are 4 marks. So 1 mark is for the comparison and 3 marks is for the explanation. Now compare was simple. That was a sort of like a described 13% less percentage oxygen saturation and same quantity of blood delivered, but less oxygen by hemoglobin to tissues, because 19.3 went down to 19.2. Now explanations for these were, I've given them in red. So the explanations were these. At high altitude, less partial pressure of oxygen in the air. Hemoglobin has a lower affinity for oxygen. In lungs, less oxygen binds to hemoglobin. So there's an increased production of hemoglobin, increased production of red blood cells, more hemoglobin available to bind to oxygen. Because of the lower partial pressure of oxygen, the body tries to compensate by making more red blood cells. That is why people who live at heights have more red blood cells. Then coming to part C, the result of the research project have led to an improved understanding of diseases such as sickle cell anemia. Now explain why sickle cell anemia is an example of a disease. So you see, example of a disease, and if you're in good health, what does that mean? If you're suffering from a disease, what does that mean? It was general and English. If I say, oh, I'm fit, I'm healthy, it means, okay, I don't have any problems. I'm not breathless, I don't have any diseases, I don't have blood pressure, I don't have diabetes, and I'm fine, I'm fit. So explain why sickle cell anemia is an example of a disease, is absence of good health. If you have a disease, well, you're not well, you're not healthy. You can't say example of a disease is because sickle cell anemia is an illness or a sickness. No, it's absence of good health. Again, I feel it's more sort of checking how well you can express yourself. Some of you can do it very well. Some of you, of course, struggle with it. So absence of good health. And number two, it produces specific signs and symptoms. Like if you have a flu, well, you're going to suffer from sneezing and fever and joints and body aches. But if you have malaria, well, you'll suffer from anemia, and you'll have high temperature with sweating and shivering. So every set of diseases had different signs and symptoms. So it will produce a specific signs and symptoms. Some diseases have a headache, some diseases have weakness, some diseases have breathlessness. As you know, we are suffering from the pandemic. Corona causes, problems in the lungs, and the person is put on a ventilator, and it's Allah's will whether he survives or he dies. So absence of good health, and it produces specific signs and symptoms, or it is having an adverse effect. Naturally, disease means that you're sick, you are unwell, maybe you cannot even perform, you can't do the normal things that you do. Maybe if you're a teacher, like I'm a teacher, I can't go to school, I can't teach. Of course, the students are going to have a lot of fun time, party time, but means that the teacher is not well, so she cannot come to school, she cannot teach you. So produce specific signs and symptoms having an adverse effect. If you have a disease where you're not well, you can't get out of bed, you can't uh, probably go and have a bath, you don't want to feel like eating anything, and you don't have the energy to do anything, and you just want to lie in bed and curl up. And depends upon the severity of the disease, of course. So explain why sickle cell anemia is an example of a disease, and that is what you were supposed to give me. Absence of good health, or something like that, meaning you could have also said reduced impaired function. You could give me some, like in anemia, less oxygen is transported. So you could give me a little more explanation about what anemia and sickle cell anemia is. Of course, it's a genetic disease. And with that, we come to the end of this video, and we'll continue the rest of the 2 questions in the next video. And thank you very much. And I hope this helps.